Okay, so you're now being recorded. That said, I do have some standard disclaimers. I'll skip to my standard disclaimer. Sometimes information or images of unpublished findings are presented here. So all presentations in AGP rounds are considered to be confidential, not to be shared outside of the meeting without the express consent of the presenters. Everything I present today, you're free to use, steal, relate, otherwise, and we'll ask the same of any other presenters. I thought it would be a good opportunity today to remind everyone who are AGP anyway. So the Academy of Genomic Pathology, which you are now all a part of, was originally established as the faculty of the Center for Genomic Pathology, which Bob Cardiff established here at Davis to have a primary educational mission in the area of pathology of the mouse and animal models of human diseases. So originally, if you wanted to be a part of AGP, and I do encourage you all to put it on your CVs, I think it's important, that meant that you agreed to and do agree to contribute content to the teaching mission of the center. So stay tuned. You all will be asked again to continue that contribution. We are getting our coursework tuned up and put back online, and will be accessible for your students or your colleagues and collaborators that maybe have no experience in pathology, but you'd like to give them some. So that's who AGP is. And without further ado, everything you wanted to know about ACE2, but we're afraid to ask. And so why ACE2? Just a brief introduction to the disease here, the clinical pathology of a patient. We'll start with histology. And I thought these images were among the best that I could find from an autopsy patient with coronavirus. And you can see this lung disease is characterized by, at this point, fibrosis, but also with acute respiratory distress syndrome, ARDS, hyaline membranes, the whole 9 yards. In this case, hemorrhagic changes and inflammation have also come in. And here's some hyaline membranes in G, some viral inclusions, we think, maybe. And we can come back to these pictures if we want to try and see if we can create mouse models that mimic any of this. In that paper, they also showed the immune infiltrates, and in a different paper, some immunostaining for 2 antibodies, one against spike protein, the S1 protein, and one against the nucleocapsid of the virus, looking at lung tissue for cells that are infected in this disease. Electron microscopy shows what these guys look like. You can see the little red arrows. These are the little viral particles inside of the cytoplasm at this point. And so I started thinking about, of course, like everybody, viral entry and what was known about that. And that's where the ACE2 comes up, which of course stands for angiotensin converting enzyme 2. There is an ACE1 as well. Was identified to be the surface receptor in the initial SARS virus, and seems to be a common receptor for most coronaviruses. And we can talk about MERS and some other things perhaps, but based on the work from the original SARS virus, this ACE2 protein and the interaction with the SARS-1 spike protein was characterized, and there was a lot of this kind of description of it. They actually crystallized the interaction of the spike protein with the ACE2 protein to understand better how it interacts, and I won't get into all the details, but the binding domains between the 2 proteins are known. And a variety of sort of partial explanations for how this all works have been given. Here's one, but the one I like better is this one. And so this one shows down here at the bottom the binding and the relative importance of the cleavage events that happen. So there's a furin cleavage that happens and a tempress. A lot of you have heard about tempress 2 being involved here, tempress cleavage. And so what appears to be happening is after this tempress cleavage, there's exposure of a part of the spike protein that was not previously exposed, and that's what is allowing the transmembrane transduction of the virus, or at least, you know, adherence, sort of permanent adherence to the membrane. And where this happens is still, I think, up in the air, whether this happens actually on the cell surface or within the endosome. So I think somewhere in binding, this can be endocytotically taken up. And then later in the process, this fusion recognition allows the transduction. At least that's my assumption. Apparently there are other mechanisms, sort of mystical, nobody knows exactly how they work, where endosomes become intracytoplasmic, and the cathepsins have been implicated in that process. Okay, where was I going with this? So a few things to know. Obviously, the first thing I wanted to know was, where is this ACE2 gene actually expressed? And I don't know if you're like me, but one of the first places feedback that I go is, here we go, is Human Protein Atlas. And so in Human Protein Atlas, you can immediately see is that the gastrointestinal tract, and here you can see it on the surface of the colon, but if we were to look at the small intestine, you can see very nice expression in the mature epithelium in the small intestine. I think this expression pattern is largely, you know, at the luminal surface, sort of towards the gut lumen, and less so in the areas of the crypts down deep where the stem cells are. And so I think this is a reasonable explanation for the finding that patients with coronavirus infection are getting a mild GI kind of symptoms. They don't typically get too severe. There are examples where more severe infections occur, but my presumption here is that because these cells are being sloughed off rather quickly, that the viral susceptible cells in the gut are not so much the problem, but a loss of barrier could still occur that would give you a secondary infection. And so there are patients that do very poorly with very severe GI symptoms, but those are uncommon. A couple other interesting places to look before I go to the lung and the trachea. The brain is interesting, and we may come back to this. So in the cerebral cortex, there is some expression in neurons. Come on. It's thinking about it. There we go. So, you know, I think it's faint, but I think there is some real expression in neurons of ACE2. And that has been described in other contexts, but here you can see it in Human Protein Atlas at some level. If I go back, go to lung, start with nasopharynx. Nasopharynx, not very impressive, actually. I don't know, you tell me if you see a cell that's positive there. Maybe up at the surface, there's a little bit of expression, sort of like we saw in the gut. That may be enough for the nasopharynx. But importantly, in the lung, you can see some detection of ACE in macrophages. I never know if macrophages in the lung are real staining or some sort of background. Oops, stay up there. And then I think you can, in some areas, and it's easier to see actually if I pick one that has fewer macrophages, I think you can see a rare cell that's positive. And so as I look around in this rather pristine lung tissue, arguably you do get an occasional cell in here that has a little bit of expression. And so based on that, and one of my collaborators I've been working with a lot came out of Barry Stripp's lab, he showed me, this paper is interesting where they took human lung tissue and they did single cell RNA sequencing of it. And they tried to ask which cell types had expression of this short list of proteins. And so at the top in the red box is ACE2. And so when they dissociated the individual lung cells, and they identify them with other markers that characterize each of the cell types, you can see ciliated cells here, and then you see AT1 cells, and this cell type, AT2 cells. Now, these are violin plots, so the way to look at these is if there's a lot of expression, like if you want to look at CD68 in macrophages, almost all the macrophages are positive, and there's a colored violin in the background here. Here you can't see a colored violin, and that's because the colored violin would be down at the bottom. So most AT2 cells are actually ACE2 negative, but a subset of AT2 cells are the ACE2 positive cells. And so this, I think, is interesting that it's actually a very small cell population in the lung of the AT2 cells that are the source of ACE expression, at least by RNA. And of course, they could express the message, and the protein could last as those AT2 cells differentiate. So then the question is, what are AT2 cells? And so that's this figure. AT2 cells are over here at the far right-hand side. They're depicted as the plumper alveolar lining cells. And the concept here is that they are the cells that are more stem-like or progenitor-like in the alveolar population. And the AT1 cells are the more differentiated alveolar cells. And so, as you might expect, AT2 cells are actually increased in number in the setting of chronic lung disease because they're trying to, they proliferate in an attempt to repopulate the alveolar lining. So all these other cell types are not really the target. It's this progenitor-like alveolar cell and only a subset of those, and we haven't figured out what's in common about that subset. We're trying to mine that single cell RNA-seq data to try and figure out what else we can say about that subset of AT2 cells. That's an area of inquiry for us. In any event, that's pretty interesting when we start thinking about modeling this disease in mice. And I'll go back to the PowerPoint briefly. Oh, I actually had that. The next question, of course, is where's AT2 in mice? ACE2 in mice, and then related to that, how similar is the ACE2 protein between mouse and man? And so you guys probably already know some of the answer to this. If you look carefully at the homology, they're not super homologous. And somewhere I have that homology. Apologies. Too much information. Where did it go? Ah, here it is. Good old-fashioned sequence alignment, and this is tailored to the places where the human ACE2 coronavirus binding sites are known to be, and where the TMPRSS2 cleavage site seems to be. And so the cleavage site is pretty similar, but the binding site between human and mouse is significantly different. So the next question I asked is, how conserved even is the human gene? And it turns out, and hopefully I can find the right window for you all, it turns out that there's a lot of heterogeneity of the human. Where did that go? ACE2 gene, there's polymorphisms, and this has been, because this is angiotensin converting enzyme, this has been studied in a lot of detail by the people doing hypertension research. And here's just one of many papers showing genetic polymorphisms that are associated with essential hypertension. The polymorphisms in ACE2 there are many that are in common between different racial backgrounds, but there's some that are unique to different racial backgrounds, which is an interesting thing to think about. And I think it's an open question as to whether different polymorphisms in human ACE2 have more significant binding, more susceptibility to coronavirus versus more resistance. And there was, in the original SARS epidemic, one study that looked at that. They only identified 2 coding sequence polymorphisms that they knew about at the time, and they did not find an association to the susceptibility of viral infection related to those. But I think that there has to be more to our thought process, and I would say we need to put this in a bigger picture context of knowing both what is the individual person's AT2 cell population number, as well as what is their ACE2 coding sequence, and is that more sticky or less sticky than other polymorphisms. So there's one other thing to know, and I think this is an interesting little side note. Where is ACE2 coded? So ACE2, it turns out, on the mouse is on the X chromosome, and on the human is on the X chromosome. This to me is interesting. We're seeing, internationally, we're seeing about twice as many deaths in men than women. And this fits with my polymorphism heterogeneity resistance versus susceptibility hypothesis, because what this means, of course, is that a man will only have 1 copy of ACE2, where a woman will have 2, and that may offer her some heterogeneity that allows better resistance. AT2 cells, for example, that express the more resistant form of ACE2 could survive better and provide more lung re-epithelialization. So I'm sure I've prepared more to tell you guys about, but I'm going to stop there and let other people share some images. I don't have my own slides from these mice yet. Kent Lloyd apparently is getting several of these mice in. Oh, I will say I do have, as an introduction, some of the original papers. And you guys have probably all looked at these, but this paper is the one where they introduced the human ACE2 gene under control of a large fragment of the mouse ACE2 promoter region. And so they get a relatively normal distribution of ACE2 expression, theoretically, and characterize that. The other paper, and I alluded to this in the invitation to you all, is the one with the K18 promoter. I would say this is sort of the most commonly used or planned to be used mouse that I've seen. And obviously there's kind of a couple of problems in my way of thinking with K18 expression of ACE2. They do show this figure where the non-transgenic and the humanized K18 ACE2 get similar sort of bronchial infection by virus. These mice, even though we wouldn't think that K18 was going to be expressed in brain, they get very severe encephalitis with the virus. There's some description about whether this is just viral cell death without encephalitis or a true encephalitis, and we can talk about that. You guys probably know, if we were to look at human expression of K18 in the lung, it's kind of everywhere. And then I won't show it to you, but interestingly, there is a difference between mouse and man in this regard, with mouse actually having less expression of K18 in lung. So here's human, and you can see, I don't know, I'll pick this one, I guess, because it's got bronchi as well as alveoli, that both the bronchial lining cells as well as the alveolar lining cells are very K18 positive. In the mouse, in my experience, and again, I could bring up an image of a slide if you wanted to see it, the expression in the alveolar lining cells goes way, way down. And so you see tracheal and bronchial expression, but not alveolar lining cell expression of K18. Okay. Open for questions or discussion, and then we'll move on to Jerry and Stephanie and anybody else that has good stuff to show us.
Sandy,
Yes.
you dealt with gender differences. What about aging? That's all.
Yeah, I think it's a good question, and I tried to look at the Human Protein Atlas, which does have the age of the patients here, to see if there looks like there's a difference in normal lung in an older person with respect to AT2 cell and ACE2 expression. And I don't really see a difference, but it is still possible that, you know, I just have a limited sample and I'm not seeing it.
Hey, Sandy, it's George Thomas here.
Yeah.
You know, so also looking at the, you know, we've all been focused on ACE2 and the lung, but actually if you look to the protein atlas, it's high expression in kidney and also highly expressed in kidney cancers, highest expression. And so, and if you look at sort of the patient population, you know, in what sort of killing them, there's a huge number of them who are actually getting kidney failure and then going on to dialysis. We don't hear as much as the ventilator need, but the needs for dialysis is just as high. And I think that sort of makes perfect sense that there's also a huge amount of infection that's going on in the kidney. And so in terms of the mice, maybe something to think about as well.
I think that's a really good point. And one of the things that we've discussed here is whether there's a role for testing urine,
right,
as we test nasopharyngeal swabs. And I think you're definitely correct. This is where you see a lot of expression here. I'm showing the tubules, not in the glomeruli, and
right.
I think it's in select tubules. I don't know whether it's in the distal or proximal, but you could
Yes,
probably
that
tell
was
me.
my question. That was my thought as well. I couldn't figure out from the TMA where it was. Or if it made a difference.
Yeah, really good point. Thanks.
Hello, Sandy. It's Raul here.
Yeah.
I seem to recall ACE2 expression in placenta. Do you know anything? Did you come across that? Because I was surprised that so far there seem to be limited reports on pregnancy problems.
Yeah, I don't know. I'd be happy to learn more about that. Does anyone online know?
There was a report, I think, in last week's New England Journal about pregnant women that were coming into New York, but the main focus was the fact that about a quarter of them were asymptomatic, but they were positive. But I didn't see anything about outcomes in terms of what their what their kids were like.
Mm-hmm.
Okay, thank you.
Okay, I think there's plenty more we could just discuss, but I want to turn it over to Jerry and Stephanie. I'll let you guys arm wrestle for who goes first.